The title of today's lecture is Computational Neuroscience, Bridging Scales with Mathematics. So my name is Gaute Einevoll. I'm a physicist by training, and I work at the University of Oslo and the Norwegian University of Life Sciences. So I'm trained as a physicist. So and this is one of the, I would say, major trends in not only in neuroscience but life science altogether, is that there are many people like me, people with a training in physics or informatics or mathematics, who join the field. So I wanted to start with a picture of this guy. So this is Niels Abel. He's quite famous in Norway. He's even made it all the way to the stamp. So he was this famous mathematician who lived at the early part of the 19th century. And the reason I bring him up here is that you can see on the stamp here that he died very young, only 7 years old. And he died of tuberculosis. And I read his biography, and on his deathbed, he was so upset about the doctors at the time who couldn't fix this simple problem. Why do you need to die of this simple tuberculosis? And now, of course, in our part of the world, we don't die of tuberculosis anymore. But now it's really if you have a sickness in the brain, if like either with, say, like dementia or some psychiatric disorder like schizophrenia, it's not so much that the medical science can do for you yet. And it hasn't been for the lack of effort. There's been lots of people working on figuring out how the brain works for a long time, and still we seem to be there's limited progress, at least in terms of I would say, both treating patients, but also in understanding how the brain works. And a typical reason that sort of that one gives for typical idea why we're making so little progress has to do with the numerous scales in the brain. Both spatial scales and time scales, they vary quite a lot from the spatial scales all the way from the size of molecules all the way up to the full human, and also in time scales all the way from like nanoseconds and even shorter, the time scales of molecular interactions, all the way up to the lifespan of a human being. So and in the course, in other lectures in the course, I think you've been introduced to this complexity. All the brain has been studied at all these different scales, and there's all kinds of complexity, and it's quite difficult to link the functions of these scales. So this is maybe a little bit discouraging. On the other hand, I like to point out that we, meaning mankind, has already been quite successful in bridging scales, because many of us now go around with these smartphones, these very complicated telephones who can, or actually small computers where you can do all kinds of practical things. And if you look at all the scales in the brain, so we have scales from starting with atom, molecule, molecular network, neuron, neural circuit, all the way up to the brain. We have very similar kind of scales in the smartphone. They both start with, atoms are atoms. There's no difference between an atom in a smartphone and an atom in the brain. And then in the smartphone, these atoms are put into special crystals, and even these different crystals are mixed together to make these tailored materials. And with these tailored materials, they are put together then in a tailor-made way to make the transistor, which in some sense is analogous to the neuron in the brain. And then you can go upward still, that the chip is sort of like a complicated circuit of these transistors, and then you put like several of these chips together with other things, and you have the whole smartphone. So we already have a system. We have bridged all these scales from the atom all the way up to the full system. So how did we do that? Well, the key thing was that in order to bridge these scales, we needed a special language, and the language was mathematics. So it's really mathematics that has made this linkage between these different scales possible. And it's not only scales actually in terms of like the building the smartphone, also programming of the smartphone. There are several layers of how to. I mean, when you program a smartphone, we can program the smartphone with our fingers because it's made that way. But actually, when you program with your finger, it's actually translated down into instructions for the transistor via a set of different programs. So even though no single person understand the system at all these different levels, we are nevertheless able as a community to make smartphones and actually make it behave the way we want. So mathematics is a key thing. So then we can ask ourselves, what's the point with mathematical models? And the main point with mathematical models is that they give more precise statements about nature than models in words. So it's not that the models necessarily are correct, but it means they are more precise because they are models that give out numbers and can actually answer questions like they can give quantitative predictions like how much, while models in words give qualitative predictions, meaning more in what directions. So what's the point with these precise models? Well, if you want to find out whether you have a correct hypothesis, ID, model, candidate model for how the world works, or a piece of the world, a piece of the brain works, if you have a model in mathematics, it's more easier to find out whether the model is right or wrong, because the model gives much more precise predictions, while the model in word is sort of like is giving more vague predictions, and it's difficult to find out whether it's right or wrong. So an example here is, just to illustrate this point, is if you're sort of standing by a lake or by the sea and you drop rocks in it. So I guess everyone has done this at some point in their life. And then you can make these observations, and you can start making, say, models in words, and you can find out, for example, that all rocks, big or small, so meaning regardless of their mass, they use the same time to fall to the sea, to drop the height h, so it's independent of the mass. And you can also make the observation that the larger the height, if you drop it from a higher height, then the larger the speed will be when the rock hits the water. These are correct statements. But the point is that if with a mathematical model, which in this case is like Newton's law, which we learn about in introductory physics courses, give more precise predictions. It actually tells that the time it takes to fall to the water is the square root of 2 times the gravity, the acceleration of gravity and the height, and that applies for all masses. And they also, from the laws, it follows that the velocity when it hits the sea is proportional to the square root of the height. So it has a more precise prediction, because it doesn't only say that the velocity increases with the height. Increasing with the height could mean that it was like the square of the height, or the proportional to the height. No, it actually says very precisely it's the square root of the height. And this means that it was easier to test whether this law or this mathematical model was correct or not. Also, when this law was established and observed for like falling rocks, or maybe even falling apples, like Newton supposedly studied, it turned out that the same model could also be used not only to understand the world at the scale of the apple, but also at the scale of the Earth. For example, the ebb and flow of water due to the interaction between our oceans and the moon, and also the planetary orbits. All this came out of the same law. So the key thing here is that these like the big breakthroughs and the big both in understanding our nature, at least, or like how gravity works and how forces work on Earth, and also in terms of engineering in making a smartphone. It has been based on a very tight collaborations between, say, two hands, the two hands of science or natural science, experiments and mathematical modeling. So it's it's the joint or the collaboration between these different branches of science, different methods, that has led to these fantastic breakthroughs. So this is something that I would like to argue in this lecture we also need in neuroscience, in particular to bridge these different scales. So that's why the sort of like the overall, say, like a slogan of this talk is neuroscience with both hands. This also means that I think just like in engineering or physics now, where the people who do experiments and people who do modeling, they are trained together from from the time they enter university. This is not the case in neuroscience, but maybe at the moment, but maybe it should be. So, and one thing to remember is that we know the mathematical equations that the brain follows at these different scales. So we have, like mankind in the 20th century, found essentially all these basic equations that the brain follows at these different scales. What we don't know yet is sort of how to use this, or we haven't been able to bridge these scales properly, how to show how the equations at one level is linked to the equations at another level. So that's one of a major challenge today. While talking about mathematical modeling, I want to point out here that the different types of mathematical modeling done in neuroscience, and three maybe main classes are shown here. The first one is sort of statistical modeling, where one tries to make like mathematical models, statistical models of which encapsulates a lot of experimental data. And then you also have what are called interpretive or normative modeling, where you can ask the question, what's the point of this neuron having these particular properties? This makes sense to ask for a system like the brain, which has been under evolutionary pressure, so it has actually adapted to be useful for the animal. This is not very useful for like dead systems. And the third one, which will be the main focus of this talk, is this what are called mechanistic or physics type modeling. For example, how does the behavior of a neuron follow from the molecules inside and outside it, from the behavior of its parts. So all these three types of modeling are useful. They have different scopes and different uses, but and they are all useful, but I'll focus on the third one. So the star of the brain, I would say, or like the key element of the brain, at least in terms of signal processing and thinking, is the neuron. So this is a beautiful picture of a neuron where you can show both the soma, the center of the neuron, different dendrites, and then also this single axon sending signal out from the neuron. And the key thing is that at this point in time, we know quite well how, at least the principles of how signals are processed in neurons. We know that in these dendrites, there's incoming signals from other neurons, which spreads electrically to the cell body or the soma, and if there's enough current coming in to the soma over in a time window, then it fires this action potential, this sharp electrical spike that lasts about 1,000 of a second, and this propagates by itself down the axon until and then arrives at the synapse, where this action potential leads to this release of signal molecules across the synaptic cleft to the receiving neurons. The key thing here is that we know we have mathematical descriptions, good mathematical descriptions for all these processes. So we have a good way to model how a single neuron behaves. However, a single neuron is not particularly smart, but at least we have, so we want to understand networks, But given that we have this good description of a single neuron, we have a good starting point for studying networks. And the starting point, I would say, from this good single neuron models was this very important work by Hodgkin and Huxley in the 1950s, where they developed this equivalent electrical circuit description of the membrane potential across an axonal membrane. So, and the model looks sort of like just a skeleton of it is shown here, where the first equation actually shows that the conservation of current. So it's actually the same kind of law that also applies for electrical circuits in transistors or in computers. And then the key element that that Hodgkin and Huxley discovered or invented was that they had these separate currents going through the membrane due to sodium and potassium ions. So they were able to find this system, this squid giant axon, where they could do all kinds of manipulations of the system and actually determine the elements and determine the model parameters. And then when they had essentially developed this model, they could make this really exciting prediction that they could sort of, with the parameters fitted one set of experiments, they could test the newly developed model on whether it could predict that an action potential propagated down this axon. And it turned out it could. Not only it could, but also that the shape and velocity of these action potentials in that the theory predicted was very, very similar to what the experiment predicted. So the difference between while the experimental value was like 21 meters per second, the theory was about 19 meters per second. So so this was a big success, one of the biggest, I would say, successes in mathematical biology altogether. And what they also did then is looking at this multi-scale perspective, the Hodgkin and Huxley, they were able to bridge the scales of molecules, or the molecular networks, and the scale of neurons. Because one thing they had inferred from their models was that there had to be these particular ion channels, which are selective. Some are just conveying sodium ions, the other ones are just potassium ions. So this was a prediction from their modeling. And later this was sort of seen that these ion selective, or these selective ion channels, actually exist, and they consist of these corkscrews of proteins that go back and forth between the inside and outside of the membrane, and together make a little tunnel for these ions. So this was a great success, both in terms of understanding the origin of the action potential, but also in terms of predicting these ion channels, which at the time could not be seen experimentally, but which has been sort of been seen in great detail. So this scheme that was used to develop by Hodgkin and Huxley for the action potential could sort of rather straightforwardly be generalized to whole neurons, not only axons. So, and that has been developed into this what is now known as this compartmental forward modeling scheme, also called as sort of like biophysical modeling. So where the idea is that you take a neuron with all its branches, and you divide these branches into small compartments, and these compartments are so small that the key dynamical variable, the membrane potential, doesn't change within the compartment. And then you make a single equation for each of the compartments, keeping track of all the electrical currents that goes into this compartment and leaves the compartment, both through the membrane and also through inside the neuron through neighboring compartments. And then you end up with one equation like this law here, this equation here, for each of the compartments. And this is the same equation that is used to model electrical circuits in silicon chips. The main difference is that in the brain, the key sort of like element that conveys the signals are ions, meaning atoms that has lost an electron, quite heavy and bulky things, compared to the electrons themselves, which are just pieces of atoms, which conveys the signal in computers. So yeah. So now when you get one of these equations for each compartment, then of course you get a bunch of equations which you have to solve together. But then if you take this, if you specify all the parameters for all current terms in all segments, all compartments, the mathematical solution is in principle straightforward. So there are now several tailor-made free mathematical simulators which are available for doing this. So examples of these are Neuron, Genesis, and Moose. And here is an example of a simulation that, or animation of such a simulation, where there are, there are like 4 or 5 neurons, which are then reconstructed, and the coloring tells about the membrane potential. And we can see when you have these flashes, occasionally you see these flashes, which corresponds to these neuron firing action potentials. So this is just to illustrate that we can model, in principle, model whatever we want at this single neuron level using these multi-compartmental or this compartmental modeling scheme. So that's for neurons. But as I said, a neuron is not particularly smart. You want to understand networks. So then that's sort of like the next step, trying to go from neurons up to neural circuits. So then question one has to ask is sort of how should I model the neuron? And it's not always the best option to use this most detailed by like compartmental model. And one reason is that it's computationally expensive. So it's sort of like with this kind of detailed neural models, the size of the networks, the number of neurons that you can include in network simulations are smaller. But there's also another thing is that it's difficult to find the model parameters of these biophysically detailed full compartmental neural models. So sometimes it's maybe easier to actually have simpler models with fewer parameters, which you can then tune to have certain functional properties. Anyway, there's several things you can do to simplify the neural model. The most obvious thing, which almost everyone does, is to remove the axon, because once an action potential is generated down the axon and starts moving down the axon, it moves typically with a constant speed. So you don't need to compute it all the time. You can just time it when it leaves the neuron, and then just compute how long time it takes to get to the next neuron, or the neuron it projects to. And then you can also simplify the dendritic structure, and all the way down to a single neuron, an isopotential neuron. And this is not as dramatic as it looks. I mean, it could be, if the dendrites are quite thick, it could still be quite branchy and still effectively be isopotential, meaning that all branches of the neuron have the same potential. One popular model, which has been studied a lot, is the so-called integrated fire neuron, where this is sort of a single compartment neuron, where the dynamics, when it sort of integrates information before firing, is described by a rather simple differential equation. It's called just like an RC circuit kind of neuron. And then if this neuron then receives both excitatory and inhibitory synaptic inputs, the membrane potential, shown in red on the lower part of the figure here, the red curve, is going up and down. And then whenever it sort of hits the threshold, shown by this green dotted line, the neuron model sort of produces an action potential, or you put it in by hand, and then you reset the neuron, and then it goes to the like a starting potential, ER, and then the whole process continues. So this is a very simple model that gives a lot of really encapsulate quite a number of salient features of how a neuron integrates information, gets information from other neurons, and produces action potentials. And it's also mathematically quite and conceptually quite tractable. So this has been a popular model to study. And if you're interested in playing with this, how like neurons of this type, how they together can sort of actually do things, we have developed this little tool called Neuronify, which is where you can sort of actually, with this plug and play kind of canvas, you can pull in neurons. So here you see that you pulled in 2 excitatory integrating fire neurons, and then you pull in actually a current source to make this to drive these neurons, and you pull out a voltmeter, so you can see how these neurons respond to this input. And here the neurons are hooked up together, and then presumably it's also, and then it's even hooked back, like, so it's like a recurrent excitation. And then we can see, let's hook up to the voltmeter, and we can see how this actually the dynamics of these neurons. So these two curves there shows the spiking behavior of these 2 neurons. One can also then obviously have more complicated circuits, just like the example circuit shown here, where the red neurons are inhibitory neurons and the blue neurons are excitatory neurons, and there's like different readouts of neurons at different levels. So this is then something which is not only available for laptop computers or normal computers. You can also actually use it on your iPhone or Android or like tablet computers. But this is more for, say, like learning educating yourself in how these neurons behave and what you can do with small circuits. If you want to sort of to study real pieces of the brain, you have to include more neurons than you can sort of easily keep track of on this with Neuronify. So then you have to have other tools, and a very popular tool for simulating many integrating fire neurons is this NEST, and this is an example application where this Potjans-Diesmann model, where they include 80,000 integrating fire neurons, which corresponds to 1 square millimeter of cortex about. And in this model it has 4 layers, sort of mimicking the multi-layer organization of the cortex, 2 neuron population per layer, and so on. And this model, which is then available for everyone to load and to download and play with. The synaptic connection rules are taken from experiments, but all these things can be changed as we want. So then with this with this kind of model, we can drive this network with visual input, or meaning thalamic input. And this is an example of a simulation where there's a thalamic input representing visual input coming in at time 900, and then you can see that there is this burst, or this strong, like transient burst of activity immediately afterwards. So each of these dots correspond to a spike of a neuron, and while it's altogether 80,000 neurons, this is only a subset of the neurons are shown here. If we showed all neurons, you wouldn't see anything. So the key thing here is that now we can make, if you have an idea, a hypothesis about how this piece of cortex does something, computes, we can formulate it mathematically and come out with very precise predictions shown with these raster plots on what the nature would look like if this model was truth, and then we can sort of compare with experiments and see and build on that and go back and forth between experiments and modeling. So then that is sort of if you only want to model spikes. But there are also many other ways to measure neural activity. If you want to measure cortical activity, in addition to measuring spikes, which we typically have done with a sharp electrode positioned next to neurons in the cortex, you can put in now all the kinds of contacts or electrodes with many contacts. So instead of measuring from one place in the cortex at a time, you can maybe actually measure from like tens or hundreds or even thousands of positions. Now these are not, because of the way these electrodes are made, then that it's not, well it's not always, it's sort of not always easy to extract spikes from them. You can also, but also you can measure the low frequency part, what's called the local field potential, which then gives complementary information about what's going on in the brain. And then you have a whole new, whole separate set of measurements based on essentially shining light on the piece of cortex and measure what comes out, record or take a picture of what comes out. And then in order to get good picture, interesting information, you have to put in different kinds of dyes and use different kinds of different kinds of techniques. So for example, this 2-photon calcium imaging is used a lot now, voltage-sensitive dye imaging, there's also something called intrinsic optical imaging, and so on. So then we have these different measurements of neural activity. What shall we do with it? Well, traditionally it's been a lot of statistical analysis on this, where they try to sort of look at how different measures are correlated and how it's correlated, different measurements are correlated with behavior and so on. And that's typically a quite difficult procedure. It's difficult to get really solid insights from that. That is at least my experience. But one thing to keep in mind is that if you know the neural activity or a neuron, all these different measurements reflects the same underlying neural activity. For example, if you measure a spike with a sharp electrode put close to the soma, you measure a weighted sum of the transmembrane currents in the soma region. While if you measure the LFP, the contribution from a single neuron is still a weighted sum of transmembrane currents, but it's not only in the soma region, it's all over the neuron. And then if you do these optical recordings, then what you measure is the weighted sum of membrane potentials close to the cortical surface. So the thing is that it's these different measures just reflect different aspects of neural activity. So what we need to do in order to make contact with experiments is to make out all this, what we call do the measurement physics. You need to work out these mathematical connections between the neuron dynamics in your model and the different experimental modalities. Because then you can compare predictions from your model with not only with spikes that you measure, but with all kinds of measurements. So one thing, for example, that we have been involved in doing is making tools and calculating electrical signals from neurons, not only spikes, but also this what's called the LFP, the local field potential. And this just illustrates that the interpretation of, if you just take a signal like the local field potential, it's not so easy to understand what aspects of neural activity it really reflects, or it's quite difficult to interpret the signal. And here we see that on the left animation here, this is the extracellular potential, or the local field potential, that you see from a neuron. This is a pyramidal neuron that gets oscillatory current input, like at the one synapse high up in the apical dendrite, and you see we get this nice dipolar pattern. And what you pick up with like EEG or outside the brain is actually the sums of like millions of contributions like this. But if we instead then not inject it with 1 hertz, inject with 100 hertz, we don't get the same thing just 100 times faster. The shape of the potential around this 100 hertz driven neuron is very different. It doesn't have this nice dipolar pattern. And it turns out that just because of this, the different measurement physics of these two situations, you see much more of the 1 hertz than the 100 hertz. So that means if you don't do this modeling, you would actually misinterpret recordings of the local field potential. So the perspective then for model testing is that if you have this candidate model on the left here, candidate model for say how a piece of cortex works, then you should not only compute the spikes, you also should compute the other things that you can measure, like the local field potential and results from voltage-sensitive dye imaging and two-photon calcium imaging, and so on. So this candidate model, mathematical model, should say predict all these available measurement modalities, and then afterwards, when you have all these measurements, you can then ask what of these candidate models agree best with all experiments. So the idea then is sort of to have your hypothesis formulated precisely in a mathematical model for a network, and do the proper mapping of this like network model to all these different measurement modalities, and then compare all these measurement modalities with all the measurements with the predictions, and then go back and forth and refine your model, and so on. So this is an example of pursuing this like program, where we have used a combination of this NEST tool for computing spikes and LFPy for computing local field potentials to compute compute from a specific network model, not only the spikes, but also the local field potential that you would measure at the same time. So this is the same one and single model that predicts both the spiking activity and the LFP that you will see when you get this, well, in this case, thalamic input at the time 900 here. So then we could sort of these you can measure with an electrode like this, penetrating the cortex. You can measure both actually, and then you can actually have a better way to find out what of the candidate models is most promising. So this is all then inside the brain. So this is something you can only do on we typically do it on mice and rats. But we would also like to be able to make predictions of what will be measured in non-invasive imaging, like the things you could do on humans. So now there's a big development that's been the last decades in terms of methods for imaging brain activity. And you can not only that you can measure tissue, you can also measure brain activity in terms of the electric activity with like EEG or electrodes like shown here, or MEG, where you measure the magnetic field outside the head, or you can measure the food consumption with a positron emission tomography PET, or such as the blood dynamics by functional MRI. And so this program of starting with network models to compute all these things you can measure should then eventually also be, or not eventually, we should also be pursued for modeling this systems level signal. So this is just an example of such a simulation. In this case, it's a very simplified model, but in this case, it's a network model that generates this virtual EEG, so like model-based EEG signals. So the colors represent the size of the EEG signal in, I guess it's microvolts. And while on the right there, you see what you would measure at the same time with functional MRI. So that's the overall program. Now, there's one big revolution which is going on at the same time. I would say it's a revolution with all these computational methods being used in biology and all this. So that maybe I'm a little bit biased, but I would call that a revolution. But there is also another big revolution stems from this the discovery the release in 2003 of the human genome, that scientists were able to map out the gross structure of the genes of these long DNA molecules in the humans, all about 3 billion base pairs, coding for about 20,000 genes or protein recipes, and also lots of all the material coding for how these proteins are going to be made and so on. But anyway, so these are now, this was a big, still is a big thing. But in 2003, it cost millions and millions to do this. But now it's very cheap. So now one can sort of take a spit sample, and you can get out your genome for I don't know, some tens of dollars or something. So that means that we can now do this, take a DNA from DNA samples from lots of people and actually compare their DNA. Not only make it for 1 or 2 or 3 people, but actually for groups of people. And that turns out to be important if you want to find out, ask questions sort of for example, if you have schizophrenia, which is a very serious psychiatric disease that affects, I think, about 0.5% of the population or something, that what distinguishes, because it's an inheritable disease, it has a large inheritability. So then you can ask the question, is there any systematic difference between the DNA of the schizophrenic patients compared to the controls? And since the DNA is the molecule is so long, you need a lot of patients to do this, to get the statistics right. So it actually took until a couple of years ago before they could actually gather enough patients and also controls and get DNA samples from all of them in order to do this statistics properly. So this came out a couple of years ago, where they've looked at, I think it was 37,000 schizophrenics and 140,000 controls, where they looked at statistically at the DNA of those people. It's 150,000 people altogether. And then what you would maybe hope for is that there was one or a couple of positions, a couple of genes that was different, or that really carried a lot of the risk, because then you can maybe have you think of therapies for actually targeting these positions, these parts of the gene directly. Unfortunately, it's not like that. It turned out that they found, I think, 108 different positions along the genome, which were sort of associated with whether you get schizophrenia or not. And that is a very difficult thing you cannot sort of, to study this with traditional biological experiments where you block a gene and turn on and off and so on is essentially hopeless. It's just too many combinations to test out. But when you looked at these genes, or when you looked at the genome at this position, you saw that many of them, these positions were had to do with ion channels. So it could be, and many of them had to do with how excitable neurons are. So now, in models, it's not hard to test many genetic variations at the same time. So I think this is sort of a key future of computational neuroscience. Is actually now to try to take this information from this big activity on genes and molecular biology and include them in our model. And I think this could be a promising and important avenue to pursue further. We're just at the start of this now. So I come to the end of my lecture. So I just thought I should give a summary of the key points. One thing is that the starting point was that the brain function relies on interaction across scales, and this makes it quite complicated to understand the brain. And then, in order to make progress, you need to be able to, to come up with candidate models, have hypotheses about how the nature may work, you come up with candidate models. But then you also need a way to find out whether your candidate model is good or bad, or whether it actually agrees with nature or not. And then it's very important to have them precise. So we need these precise candidate models for this what's called hypothesis falsification, if you want to say it with nicer words. And the key thing is that the model, the mathematics is the most precise language we have. And then as a consequence, the models in mathematics are more precise than models in words. So this is why we need to take into, to include this mathematical modeling and computational neuroscience and to make this tight, much tighter collaboration between, I think, experimentalists and people doing mathematical modeling. And I think this is for the, maybe like for the next generation of neuroscientists in particular. And what we into doing this, what we call neuroscience with both hands, and try to pursue the same kind of program that I think has been very successful in like engineering and say physics. It's going to be harder to do it in the brain to pursue the program, but nevertheless, I think it's one of the a hope for bringing brain science a significant step forward. So with this two-handed neuroscience, we need then again, just to summarize the key point here, that you need to formulate a hypothesis in mathematics, and then you need to take advantage of all the experiments you have. Experiments in neuroscience is always going to be a limiting factor. So you need to compare these model predictions with everything you can measure. So you need to be able to link your candidate model, which typically is maybe formulated in terms of, at least on the network level, in terms of neurons. You need to be able to link your candidate network model to all the different measurement modalities, not only the spikes. And that was what I planned to say.